Good morning, everyone. How are you doing today? That's Kelly Long, County Supervisor, and recording. I apologize for that. This is Kelly Long, Ventura County Supervisor. I was transferring from my phone to my computer and it totally glitched. So thank you for joining all of us today for the Santa Clara River Watershed Committee. I would like to ask if everyone could put their name in the chat box and who you were with. That would be wonderful. We've got about 17 on today, so that's wonderful. Got some good topics. So next up, we will do, if we only have 17, why don't we go around and just say hi? So Kelly Long, Ventura County Supervisor, and then I will pick Brenna, and then you can introduce and then pick someone else, and we'll just go that way. Oh, we can't hear you.
Now?
Now we can.
Talk about that. Brenna Human from Tree People. I am here with Amanda and Peter.
Okay, Amanda.
Hello, Amanda from Tree People, and we also serve as the Watershed Coordinator for the Safe Clean Water Program in the Upper Santa Clara River Watershed. So that's LA County Measure W. Thank you for having us.
Thank you. And Peter? Okay. How about
Peter Schoenbarger, City of Ventura Public Works, Stormwater.
Great. Brian?
Brian Miller. I am with Supervisor Kelly Long.
Beall?
Bedawi Muda, City of Oxnard, Stormwater.
Ken?
Did you say Ken?
Yes.
Hi, I'm Ken Schiff from the Southern California Coastal Water Research Project.
Okay, Jody?
Hi, everybody. Jody Schweitzer. I'm the Water Program Director with the Farm Bureau of Ventura County.
Evan? I got you when you weren't looking.
Evan Hobson. I'm with UC Santa Barbara and also representing the Santa Clara River Conservancy today.
Okay, Nicole?
Good morning. I'm Nicole Collier-Yamagiwa. I'm with Rincon Consultants. We work on the Santa Clara River doing the TMDL and monthly outfall.
Desirae?
Hi, I'm Desirae with the Ventura County Resource Conservation District.
EJ?
Hey, good morning. EJ Remsen with the Nature Conservancy.
John?
John Bell. I'm with Ventura County Agriculture Weights and Measures.
Rick?
Good morning, everyone. Rick Vasilopoulos with Santa Clarita Valley Water Agency.
Chuck?
Good morning. I'm Chuck Carter from the Channel Islands Neighborhood Council Clean Water Team.
Okay, Monique?
Good morning, Monique with the City of Ventura, Ventura Water.
Dana?
Hello, good morning. I'm Dana Slomiak with the Nature Conservancy.
And then I had Sakata.
Hi, I'm Brianne Sakata with the California Department of Water Resources.
And it looks like Brooks is getting on. And then we have, of course, Heidi, our wonderful watershed coordinator.
Hi, everyone.
Okay, so it looks like we got everyone. Is there anyone we missed? I thought we might as well do a little personal touch since we don't have a forum to be with each other today. So we're going to move. I'm going to pass this off. Thank you, everyone, for your time. We're going to try to keep this on a timely manner and get this going. So I'm going to pass this along to Heidi for our community updates.
Yeah, we just want to open up the floor. Does anyone have any updates they would want to share? Any upcoming programs or plans, projects? No. Yeah, Brooks. Jody, did you want to say something?
Sure. I just have a really quick update to share, and that is that on September 28th, just last month, the new ag order was adopted. So there's going to be a lot of changes to the regulatory program that applies to irrigated agriculture. And we're still, in the process of absorbing what some of those changes mean, developing the programs to help growers implement them, and really kind of starting down this road. But it's going to be a long road.
So quick question for you, Jody. The new requirements for ag, are any of them lining up with our currently existing requirements so we can kind of work together?
This was a big focus of our comments, was, stressing the importance of this regional collaboration, providing opportunity for regional projects and collaboration between ag municipalities and other dischargers. The letter of what's adopted sounds like it might be a little bit difficult, but we still can work with the regional board to try to figure out how we can implement this, how we can streamline and optimize between our programs. So we're still not exactly sure, what the interpretation or what the options available to us are going to be, but we're definitely going to continue to push hard for that.
Good.
Jody, when was that adopted?
It was adopted on September 28th.
Okay, thanks.
And Jody, since maybe I can say it, so they did not choose what we were asking for, which was option 2. They gave us some of the clarifying wording, but completely didn't incorporate some of the things we wanted, which was really unfortunate. And hopefully they'll be able to modify it a little bit more as it goes through. Did I sum that up a little bit?
Yeah, the timelines are really tight. That was what we were asking for, was timelines that would allow for opportunities for regional projects. And they're so tight that we might not be able to get there, but we might have options for custom approaches. So we're going to continue to try.
Thank you.
Thanks, Kelly.
Okay.
Okay, I just want to really quickly say, I know we're meeting virtually today, but that was just because the venue we normally use is having an event today. So it is slightly different this month, but future meetings should be back to the hybrid format. Did anyone else have a comment?
Yeah, can I say something? Heidi?
Yeah.
I was fiddling around with my camera when I first got on, so I got distracted. But my name is Brooks Englehart. I'm the district conservationist with the NR Natural Resources Conservation Service here in Ventura County, and do a lot of work with the RCD and the Farm Bureau. And we have the Farm Bureau program, EQIP, Environmental Quality Incentives Program. There is a sign up deadline of November 3rd. It's open to all farmers, ranchers, and also forest land managers, people with oak woodlands, riparian forests. So that's coming up November 3rd. We have also the National Water Quality Initiative, which is that the special funding for water quality for irrigated cropland in all of the Callegas watershed. That's also available again this year. And then we have a new program under the Environmental Quality Incentives Program that's under the Inflation Reduction Act. That one is funds several practices that are for climate mitigation, for greenhouse reduction, and that is an ongoing program. I don't know the deadline, but it's up in February or March. So anyway, so those programs are available for any farmers, ranchers, or people who manage woodlands. So you feel free to send them our way. We also have I've been working a lot with Emily Nye with the RCD. There's the RCD, and of course y'all know Heidi, that you have these new programs with the CDFA. So we can in a lot of cases there's potential we can use the EQIP dollars and the funding the RCD has kind of jointly on some of the properties, especially on some of these projects that might be really costly for the farmers. And I can get some additional funding help as well. So that's my report. Thank you.
Thank you. Brooks, if anyone was interested, could you put your email or contact in the chat? So if anyone has further questions.
No, I'll do that. Thanks.
Okay, last call for any updates from anyone. All right. Our next presentation is going to be from Kevin Schiff from the Southern California Coastal Water Research Project. Kevin, are you ready to go? Yep, you sure are.
Hey, everybody. Thanks for inviting me to your watershed meeting today. When Heidi first contacted me, she wanted me to talk about our some of the research we've been doing on trying to assess the effectiveness of stormwater best management practices, or BMPs. And so I put together kind of a 3-part presentation. And the first part is just a little bit about my organization. So many of the organizations on the line today we've worked with, but not everybody. So I figured I'd give you a little primer on who we are, and then a little bit about our project on adaptive watershed management. And that kind of sets the context for the BMP effectiveness tool, which I'll just go ahead and demo for you. So it's going to go kind of from more general to more detailed as we go through the 3 parts. But yeah, feel free to interrupt me at any point and ask questions. So Heidi, you're seeing my slides okay, and it all looks good? All right.
Ken, I am seeing your slides, and if anyone raises their hand or wants to say anything, I'll be monitoring like the chat.
Oh, that'd be great. Yeah, because I'm not looking at the chat window at the moment. So, all right. Well, if you want to know more about us, go ahead and just go to our website there. Everybody calls us SQUIRT as an acronym, which is just way easier than our very long name. So, so a few things about us. We've been around for over 50 years. We're actually a Joint Powers Authority, or a JPA. And if you don't know what a JPA is, I'll be glad to explain it. But our primary mission is to do the unbiased research to look at the effect on the environment of both human activities and natural variation, you know, and try to make the connection there. And then you know, we're just a research agency, so we don't do enforcement, we don't make policy, we don't advocate. We just give all that information to these very high level decision makers. We try to generate scientific consensus amongst all the scientists and engineers, and then provide that unbiased information to these very high level decision makers. So hopefully they'll make some well-informed decisions. So to kind of give you a sense of who our joint powers are, these first 4 are kind of the very large wastewater agencies in Southern California. You know, these next 4 are the very large stormwater agencies, you know, MS4 agencies. And then these 6 on the right are kind of the, you know, the regional boards, you know, the state and federal regulatory agencies that kind of oversee the 8 on the left. So it's really kind of, our board meetings are very interesting. So the main thing is that the high level folks, right? Like we have the chief engineers at the treatment plants, right? We have the head of the, like in the case of Santa Clara River, which is split between LA County and Ventura County. So we have the head of watershed protection as our commissioner, and the head of the watersheds and the deputy of LA County Public Works are on our board of commissioners. And then at the LA Regional Board, it's the executive officer and the assistant executive officer are commissioner and alternate commissioner. So that gives you a good sense of who gets our direct information from our research. And so I'm really glad to share it with you today, because talking to you is part of our mission. As an organization, we're not all that big. We're just about 50 people. We're almost all scientists and engineers, right? We're all researchers. And we've got, you know, chemists and toxicologists and microbiologists and, you know, all of the different ologists that you can think of. But we really organize ourselves around research themes or issue areas. And so I'll let you just kind of look at those and go, yeah, I care about that. I would say for the folks in the in the Santa Clara River watershed, probably all of these are a concern at some point. So I'm going to be talking about the stormwater BMPs today, but feel free to ask me about any of these. The type of work we do really is applied. And I talked to you, I kind of mentioned briefly about doing, consensus building. That's a big chunk of our job because those high level commissioners, they're always interested in the science that we do at our agency. But it really makes more of an impact if I can walk up and say, yeah, Squirp and 5 of the leading experts in the country all believe this is the conclusion. I think it's much more impactful. So hopefully kind of the stuff that I will do with you today will help build towards some of that consensus building. One of the other things we do is a lot of monitoring or monitoring design. And that's kind of the focal point for the project that Heidi asked for today. So let's jump into the project. It's this thing that we call adaptive watershed management and building monitoring programs to help inform adaptive watershed management. In the end, adaptive management's a great buzzword, and you guys in Santa Clara River have your own water quality improvement plans. You spent a lot of time trying to do it. The main thing you need to know is as much effort and interaction and back and forth that you guys had creating your water quality improvement plan or watershed management plan, whatever you call it. Not everybody thinks of adaptive watershed management the same way. Next watershed over, next regional board over, next county over, they think of it totally different. The state, and particularly in your regional water quality control board jurisdiction, they have some guidance on how to put together your management plan kind of guidance. They have reasonable assurance analysis and those kinds of things, which you're all familiar with. But there's almost zero guidance on how to adapt, right? All of those watershed management plans say adaptive, but what are you adapting to and how do you adapt and those kinds of things? Almost zero guidance. So to kind of give you a kind of a conceptual model or schematic of what our project was trying to do, a typical watershed management plan, right? You'll have some plan, you'll have a bunch of goals in place. Each goal will have at least one or more actions that you need to implement. And then you'll do a little monitoring and you'll say, was it successful? Have I achieved my goal? Yes or no? If it's yes, cool, keep going. If it's no, then it's okay, I need some alternate thing that I'm going to do. And then you circle back to implement that action, and this just becomes a do loop. So the goal of this particular project was on this part of the flow chart and helping you make that decision of has my action been successful? Has it helped me achieve my goal? And so, and this is where there's almost zero guidance. And I don't know how much time you spend talking to different municipalities within your watershed or between different watersheds, but pretty much everybody has a different kind of very different looking monitoring program. So that's what this was designed to do, was help people design their monitoring programs. And I guess before I go any further, does this conceptual model make sense to you guys? So I put this together and I was like, I don't know if this makes sense to anybody but me. So this is where I could use an emoji or somebody saying yes. Okay, thank you, Kelly.
Pretty clear.
All right. All right. So so this project basically had 5 tasks to try and come up with some of this guidance for monitoring. And you know, I mean, we're a research agency, so we had to create some type of people who actually have to do this kind of work to tell us what it is, and then we go through the stepwise process of defining what it is they want to know and what they're going to do with the answer once they have it, how to design a monitoring program to answer the question, and then develop a bunch of support tools so they can get down to the nitty-gritty, like how many samples, how many sites, how much data do I really need, when I look at the data, what does it mean, and then these case studies. And this is where the BMP thing is going to come in. Before I dive and give you any technical details, I want to talk about the advisory committee. So if you're involved in municipal stormwater, this list of people will look pretty familiar with you. So, you know, we've got regulators and regulated. We have, you know, Northern California, Bay Area, and Southern California. We've got phase 1 municipalities, phase 2 municipalities. We have NGO. So a good cross section of level, sector, geography. We were trying to get a statewide picture on this. So probably the one that you guys are all familiar with is IVAR. So the key to all of this is the questions. I won't go through all the details of how we come up with the questions, but if you guys want to know, I'm glad to tell you. The main thing you want to see is probably what were the final questions to build monitoring programs around. And so we'll see if these ring true to you. Hopefully they will, because they're supposed to be the 3 that almost anybody would ask in an adaptive monitoring program. Something about trends, right? I implement some action, and then do things get better or worse or stay the same? The second would be something about, you know, which is my worst site? So, you know, so where do I have to focus my effort? So something about mapping or doing some hotspot analysis. And then the third one was about how effective was my BMP, or my best management practice, right? So if it's getting worse and I find my hotspot and I put in a BMP, is my BMP actually working? And so this is this one about BMPs is the one that we're going to focus on today. So now I got to know, do these 3 ring true to you guys? Would these be the 3 that you might ask if, you know, for your management plan?
Yes, Ken.
All right. Thanks, Peter. That's reassuring.
I got to ask you a question, though.
Mm-hmm.
So there was a lot of regional board representation on that previous slide you had noted. Thank you.
Yeah.
What was the regional water board representation on, what was their opinion on those questions? Because when I read a permit, I kind of think that it's a given that the agencies need to already be doing kind of the steps to apply adaptive management. But was that the same opinion or stance that the regional boards were taking, or were they acknowledging that there is lacking guidance and therefore it might be the reason for lack of progress towards adaptive management, or even fulfilling adaptive management requirements in WDRs and NPDES permits? Does that make sense?
Yes, it does. So I'll start with the first part, which is the regional boards, you know, so the regulators and the regulated parties on this list, as well as the NGO, all agreed that this was their top 3 questions. So it wasn't a tacit consent. It took us multiple meetings and tens of hours to kind of come up with this list of nearly 100 questions and whittle them down to these top 3 that anybody in the state would ask. So it was an active pursuit, not a passive or tacit pursuit. So that answers your first question. And then the second part of your question, I'm actually going to reframe and say, you know, the state held a workshop back in right before the pandemic and said, what are our biggest kind of hurdles towards, you know, accomplishing adaptive management? You know, at that point, you know, a lot of the EWIMPs were just being kind of approved, developed and approved, or not approved, as the case may be. And they all recognize that one of the biggest challenges once they get the watershed management plans approved is how do you know if you've been successful or not? And the big fear was that from one side, right, from the regulatory side, was that people aren't going to do enough. Let me rephrase that. From the regulated point of view, on this committee was, dang, you're making us do so much monitoring. It's just such a huge effort. It's a lot of resources. It costs a lot of money. Whatever money I'm spending on this, I'm taking away from implementation of actual stuff that fixes things. Why are you making me spend so much money? And then from the regulator point of view was, how do I know if I need to have confidence in your monitoring program that you're actually making a difference, because that's what I'm being held responsible for, is going, yes, the regulations we've done, so, you know, have been able to demonstrate a market improvement. So they were both uncertain, but from different points of view. And, you know, everybody's biggest fear is that they make them do a bunch of monitoring, and then either the monitoring was too much, cost too much, and didn't need to do that much, or it was insufficient, that we wouldn't actually get an answer to the question of whether it was effective or not. So, and so that's why we needed to go through, design the program so it was fair and equitable, and that it would actually get to an answer. And so the BMP effectiveness is a perfect one to talk about. Because on all three of these questions, we did some case studies and developed some web applications that any of you can use. They're publicly available. And just before I show them to you, we had a couple of philosophical things, which is everybody should be able to load their own data into it, right? Because every watershed is different. So it doesn't help for me to tell you data. You should be able to put your own data in it. And to make sure, I say this explicitly, none of the web apps save any of your data. So it's not, they're big brothers, not, this isn't a sneaky way to get your data. So it's all, you put your own data in, and nobody's saving it. So whatever visualizations we use, they should be pretty simple and straightforward. You know, so basically non-scientists or non-engineers can actually interpret it, make sense. But then you can quickly dive to details if you want to, for the people who are, you know, scientists or engineers. It's open source, so people can just download any of the scripts to see how things are actually calculated. And this is where you can go to get them all. Here's the actual final report we went to, which you can find here on the QR code. And here's the link to all the individual apps. So I'm going to show you the BMP Performance app. Heidi, how much longer do we have for me to do the demo?
You have about half an hour.
Oh, okay, more than I had assumed. So I'll do the BMP app, and if you guys want to go through any of the other apps, I'm glad to. So, but this is the one that was requested. So I'm assuming you're looking at my browser now, yeah? Okay, great. So they all have a landing page. It looks something like this, gives a little bit of background and some links to the actual final report, and you know, so just typical landing page stuff. But this is the first place to go to, which is where you upload your data and try to answer that first question about how's my BMP working. And so I'm going to look away from the camera for a moment and go, all right, so the first step is to enter your data. So you can manually enter your data. You can download a template and upload like an Excel file if you want. But you can just upload your own data. So the first thing you do is put in what your influent concentration to your flow through BMP is, something about your effluent concentration from your BMP. Pretty straightforward. If you wind up entering a bunch of data, you can download that data set to an Excel file. And then the next key thing is, you need to enter some type of water quality threshold. And this is fundamental, and I'll tell you why in a second. The main thing is this is starting to get to some of those fundamentals, right? It's pretty straightforward and easy to use. It's flexible for loading your own data or however you want. And it's specific to you because you get to put in your own, whatever your water quality threshold, whatever your water quality goal is, you can put it in. It's kind of independent of any specific pollutant. So that's kind of nice. So the key is this graphic right here. Can you guys see this graphic all right with all the different colors? All right, great. So let me spend a moment explaining it, and then it'll make more sense. Okay, so most people, when they do like how effective is my BMP working, it's something about percent reduction, right? From influent to effluent, there's some percent reduction. I assume that's how most people do it here. Somebody shake their head. So, right. And so that works great, right? If you, have a influent that's, well, we'll just say 50 micrograms per liter, and it goes to 49 micrograms per liter, that's a 2% reduction. But if it comes in as an influent at 2 micrograms per liter and it goes to, you drop it by 1 microgram per liter, so, well, that's a 50% reduction. And so it's just an inherently flawed approach. It's easy to understand, it's just flawed. And so so we put together a graphic that kind of compares the influent concentration to the effluent concentration on the X and Y axis. And so that's the first thing, is we're relating influent and effluent, not by percent reduction, but relative to this one-to-one line, this diagonal line, which is the one-to-one line. The way we look at it is, if you're to the right of this line, right, your influent concentration is less, your effluent concentration is less than your influent concentration. You're reducing concentration somehow. That's good. You definitely want to be to the right of this diagonal line. That's a good thing. If you are to the left of this line, that means your effluent is more concentrated than your influent. You're actually adding pollutants. That's bad. So that's the first thing. The second thing is the vertical and horizontal lines. That's actually this water quality threshold that you put in, that you entered up here. And so where you want to be is to the right of this diagonal and below your water quality threshold. In this particular quadrant where green's performing well, the influent is coming in above the water quality threshold. And the influence coming in above the threshold and the effluence going out below the threshold. This is the quadrant you want to be. So if you're on this opposite side, the influence is below the threshold and your effluence above the threshold, this is a bad thing. You're making things worse. And so we just call this a fail. right? And there are BMPs that do this. So I won't ask anybody for their example. So you want to be on the lower right, you do not want to be on the upper left. And then there's some intermediate ones like this one right here. Well, we'll just say this one here, performing in the blue. It's you know, it's definitely you're getting some reduction, some treatment, but you know, it came in below the below your water quality threshold to begin with, so that's okay. You know, that's good. That's what you want to see. There's this one over here in the yellow where you're to the right of the diagonal line, so you're getting some treatment, but it's just not enough to get you below the water quality threshold. So it's performing, but it's not performing optimally. It's underperforming. So, and you can kind of say the same thing over here, where you're getting some. Anyways, hopefully this made sense. If this doesn't make sense, say something right now. Perhaps I did not do a great job explaining it.
Hey, Ken, thank you. Thank you for this. I'm an engineer, so I love this stuff. The one area that I am not clear on is where do you get your water quality standard or threshold?
So that usually comes from whichever watershed you're in, right? You either have like a water quality objective you're trying to meet, a TMDL target you're trying to meet, you know, some type of waste discharge requirement that you have. So this could be concentration. For what it's worth, it could also be load. The same structure works.
So this is something agreed upon with the regional board, be it a TMDL or, you know,
I could
alcohol concentration or
right. This would be something that's probably in your permit or in the basin plan that you're working in.
Okay.
You might have a site specific objective.
Okay.
This could be an action limit if you've got like a construction or an industrial site.
Yeah.
Something like that. So that's the beauty, is that, you know, if you look at just one pollutant across many watersheds or many regional boards, they'll have a different water quality objective.
Okay.
And so that's why we wanted to make it flexible. People put in whatever they want it to be.
Okay. All right. And is all this driven by the ocean plan? I mean, eventually, right? These are waters of the United States and everything ends up in the ocean, right?
It could be if people are really worried about downstream effects, or they have a BMP that discharges directly into a salty water body. It could be, but it could also be an inland surface water plan if you've got one of a designated water body. It could go to anything like that. So
Sounds good.
the main thing is that in most watershed management plans, and especially the ones that have this reasonable assurance modeling, they're trying to predict what their BMPs are doing in their model. And so theoretically, if you guys, I don't know if you guys are doing RAA, reasonable assurance analysis, as part of your watershed management plan, but theoretically, every BMP has a target kind of treatment that it's supposed to be achieving. And that's what this is supposed to be quantifying for you. So, and the reason is because depending on which quadrant or which color you're falling in, you might do something different, right? If you're here in the green, even this blue, you're like, awesome. I don't have to do anything else. My monitoring is telling me my implementation strategy is working, right? If you're in the yellow, you say, it's working, but it's working suboptimally. I should probably go something, you know, like maybe I should do some maintenance on this BMP, right? There's maybe something I can do to get this BMP to work a little bit better and move me from the yellow to the green.
Yeah.
Right. Yeah, Peter.
Was this developed for like sites, or was this developed for WIMPs? And the reason I ask is, hey, in Ventura County, like 10% development, it's really hard to predict receiving water quality, just because, you know, 90% is unmanaged of the runoff in open space, basically. And so it makes a lot of sense, this type of model, for like industrial permitted facilities, or facilities that, you know, influent and effluent is really well understood and is related to a discharge, or like a giant project at the bottom of the watershed. Does that make sense?
Okay, so this would be applicable to any place that you've installed a BMP, in this case, a flow-through BMP. And that could be at an industrial site or a construction site, as you suggested. It could also be at lots of municipal sites, right? So, and that could be a large regional something at the end of the watershed, just as you described. But a lot of the municipalities are moving to low impact development, LID features, or green infrastructure, right? So swales, impoundments, you know, any of those kinds of types of BMPs, and they'll work equally well there. So
that's what I wanted to hear, is that it was developed and it seems to be effective across multiple types of BMPs.
Correct.
Okay.
And when we get to the next tab, I'll be able to explain it to you even better. The main thing is that depending on which color you land in, there's actually a description of what you would do. What's my adaptive management action I would do, right? So if I'm in green or blue, I'm good to go. If I'm in yellow, I probably need to check on the BMP, maybe do some maintenance. If I'm in the red, I got problems. And so now you have to, you'll go check on it, do some maintenance, see if that helps. But you're probably at that point assuming there's something wrong with your BMP, right? It's not designed correctly. It's not constructed right. There's something physically wrong with it. So you have the wrong media in it, something that you need to actually fix. So that's the beauty of this adaptive management and this tool. Is it's not just asking the question, it's giving you something to quantify, and then a logical next step. Once I have an answer to my question, what am I going to do with the information? Yeah. So anyways, once you've entered the data and your water quality threshold, and you understand how this kind of little paradigm works, then you can just plot it up. This automatically plots your data. Here's the data that we entered above. Like here's the one that I plotted, a 20 microgram per liter influent and a 5 milligram per liter effluent. It lands in the green, right? Here's the 1-to-1 is the blue line. Here's the vertical and horizontal lines, right? Which you remember I put in as a 10 micrograms per liter. And then this tells you these are essentially different. These could be different storms. For what it's worth, it could be different BMPs as well. If you put like some distributed features, you could see which one is doing better or worse than the others. So, and then it gives you some type of overall rating, and then you can download the results, you can download the plots, and put them into a report if you want. The main thing is if you add more data, you can replot this and see how your specific BMPs are doing in your watershed for your storms. So are we liking this? Yes or no?
Yes.
All right. Well, let's pretend that maybe you're in the yellow or maybe you're in the red and you're like, dang, I got to do something. And one of your first questions is it actually something wrong with, maybe you go and you check the construction and the design specs and you're like, yeah, no, I built it right. And water's getting in and water's getting out. And maybe it's just you picked the wrong BMP and you want to know how well is my BMP working compared to all the other BMPs of this type. And so we created this third page in the app, which allows you to compare the performance of your BMP to everybody else's BMP. And so we pulled all the data from the International BMP database. If you've never been to the International BMP database, I I recommend it. It's kind of a fun spot to go. We actually updated it with a bunch of data specifically from California. And the way this works is, you can pick a pollutant. You can pick a type of BMP. You can enter your influent and effluent concentration. And it prepares these two graphs down here. And so let me just spend a minute. This is probably the most technical of all the things I'm going to talk about today, but I'll try to make it in an understandable way for the folks that aren't, scientists and engineers and statisticians. So the first thing we do is we plot the influent and the effluent against each other, just like we did in the last one. So this is no different. This dotted line is the one-to-one line. And if you remember, we all want to be on the right side of this line where we're getting treatment, not on the left side where we're getting additions. So treatment concentration reductions are happening on the right side of the line. And this big yellow dot is your 105 versus 15. So that's where this yellow dot occurs. And all the black dots are all the data from the International BMP database. And so you can see, yeah, this type of BMP gets a lot of treatment for copper in biofilters. And each of these lines is called a quantile regression. Think of the blue line as just a typical regression. It's just an average of all the different points. And then each of these other lines are basically the same thing, but just not the average. It starts around the 5th percentile, it ends around the 95th percentile. So these are just same thing as a regular regression, but just around different percentiles, not around the average. And so, so what you can do at that point is, so you can kind of just visually see, yeah, I'm something around the middle, and I'm certainly no better or no worse than all the other black dots on here. Maybe my influence is a little bit higher than everybody else's because I'm way out here past 100. There's not a lot of black dots past 100. So dang, I got some really bad influence, but hey, my treatment's okay. So it's behaving like everybody else's. And so if this is your problem, then if this is your problem BMP, then yeah, my BMP is working. So, and working relative to everybody else. The key is this graph on the right. And so all this is a probability plot. And so it's your probability of things occurring. So it's not that hard to understand what this is. And this is the effluent concentration. What it does is it takes this 105 as your influent, which you put up here, and all it does is draw a vertical line up and down, like through here. And that's how you get the different probabilities. And at this dashed line right here is what your actual effluent was. And so the way to read this type of a plot is go, okay, this dashed line hits it around 65% probability. So 45% of the time, other BMPs have higher concentrations than you. And 65% of the time, you're worse than them. So you're right around the middle, which is kind of what you're getting over here. So this is super useful, right? If you're way out here towards this tail, you're not doing very well. And if you're way out here by the 10 or 20% probably, you're doing fantastic, better than everybody else. So, so this is the tool that tells you how am I doing compared to all the other BMPs of this type, anywhere in California or anywhere really in the world. So did this one make sense? Did I explain this sufficiently? So yeah, if you're out here towards the right, you're like, oh man, I'm doing a retrofit. I'm just hosed. Something went wrong. So if you're down here, then it's not my BMP. I need to do some other type of activity. I need to go do some source tracking now. I need to, I need to go put in a treatment train to fix something else. I need to do some flow control, something, but it's not about that my BMP isn't working. So
Ken, just a clarification.
Yes.
As stormwater managers, most of us responsible for budgets and so forth, we're going to be thinking about BMP implementation, efficiency, and cost. Don't forget cost, money. So in this graph number 2, your probability of effluent concentration at influent, when you see these are other BMPs, is the objective to play on the right side and picking different BMP types?
Oh yeah, so you could do that. You could go up here and keep the same influent and effluent concentrations and pick different BMPs.
Yeah.
That's certainly a feasible way to look at it. Another way to play it is you could your influent concentration, you can stick in what effluent concentration you want to achieve.
Yeah.
And see what your probability of achieving that is.
Okay, with the different BMP types. Yeah.
A third way to use it, and I haven't seen too many people do it yet, but one I would recommend, because I'm into the monitoring and the adaptive management piece, is when people are doing their RAA models now, their reasonable assurance models, their watershed models, and they're plugging in BMPs, they pretty much just stick in a % reduction. Just, they just stick a number in there. And it almost always, they never put any uncertainty estimates on that.
Oh.
They just put in a single number, say it's going to reduce whatever, 10%, 20%. This allows you to say, for my influent concentration and this type of BMP, here's the actual amount of uncertainty that I should be plugging into my model to say, if I do this strategy in the model, what's my actual likelihood of achieving that?
Yeah.
And coming up with a probability of success. That's what I think would be extremely powerful. And if you're thinking about implementation strategies, that'll save you tons of heartache on the cost side, right? Because when something's super expensive, you want to have a fair amount of confidence it's actually going to achieve it.
Yes.
And so I think using this for uncertainty bounds on your modeling would be really powerful.
Agreed. And I think the regional board may be appreciative of that.
I would hope so.
Yeah.
Hey, Ken, so you have about 5 minutes left.
Great, because I'm going to go to my very last slide. So, of course, it's a powerful tool, as you can see. Hopefully, it meets a lot of, it answers a very specific and important management question. It's somewhat simple to use, and at least the first one is somewhat easy to understand. So, if you're not a researcher, it still makes sense. You can dive into details if you want. If you are one of those, really detail-oriented person, and you want to know exactly how it works, and you want to download data and upload data, you can do all that. But of course, we're not done. As researchers, we're always keeping stuff going and trying to make it better and advance it. So we are advancing this BMP. We're adding more data to the comparative tool. So more types of pollutants, more types of BMPs, more storm events for the pollutants and BMPs that we have, just trying to build out that data set a little better. And so part of that is a regional BMP monitoring program. Ventura County Watershed is part of the Southern California Stormwater Monitoring Coalition. So Heidi, I don't know if you are super familiar with them, but that might be a good topic for a future watershed meeting, is to have them talk about the SMC, the Stormwater Monitoring Coalition. So, and they are designing a region-wide monitoring program for BMPs, and they're good. That's one of the things that's really going to beef up this tool, and specifically for Southern California, so it's directly applicable to you. The second thing is we're building a tool very much like this for hydrology, because I'm sure for your watershed management plan, a lot of it is on capture or capture and use. And so, so we're building them for hydrology now, something analogous, but specifically for flow control and volume capture. And then, of course, mashing up the flow and volume with the water quality. So, and then the last one we're working on is building an app that'll help you not just tell you if you need maintenance like the one we have now, but when it'll actually start forecasting when you're going to need maintenance on your BMP, which will be, you know, right now, I'm sure for who's ever got BMPs right now, you kind of, if you have any maintenance program at all, it's on a very static, you know, once a year or after the first storm type of a thing. It would be nice to have something that where you could predict and say, you know, after the next 2 storms or the next 3 storms, I'm going to need maintenance, you know, or after this amount of cumulative rainfall, it's going to need some maintenance. And so it's more, you know, or even real time, you know, gosh, it's been working great, but after the last storm, the performance dropped by 20%. It's time to go out and fix it before it drops anymore. So that's where we're at right now.
Oh, thank you, Ken. Does anyone have any last questions?
Can you share that QR code so I can see it again? I'm
Oh,
having
yeah.
a hard time finding it on the website, Ken. So.
Oh, okay. I will. This is what I'll do. Here, let me just re-show this.
Or just drop the exact link in the chat, and then I can find it. Because
okay,
your website's great.
And also Heidi
I'm
can
also
send
not a
that out
wizard
to
now.
us.
Yes, I'll do all of those.
Yeah, when I post the recording of the meeting, I try and include all links and web pages and contacts mentioned.
Great. And are you going to have a copy of this presentation? I have people in the organization who are interested in the city.
Yeah, Ken, could you send me your presentation and I can post it as a file underneath the recording?
I'd be glad to.
Okay, awesome.
Thank you, Ken. I'm really looking forward to playing with this. Personally, I would love to play with this as much as possible.
Great.
I'm like a kid in a candy store with this stuff. So
hey, do me a favor, would you, after you play around with them a little bit, drop me a note and let me know how it worked out, because we're in constant quest of improvement. So one of the things we don't want to do, or we're afraid of doing, is putting these types of tools online and then you know, make them not being maintained.
Yeah.
So
we want to make sure they're functional if we're going to leave them online.
Cool.
Well, and I would like to say thank you to Ken for all the research and background you had to do to get to this point, and also the capital to build this modeling system that we could all use, in which you're saying that we can use for free, which is very nice. I know it's very technology driven, so that's good when we're making those types of decisions. And I know that we're all looking at how do we improve things. Not everything in Ventura County is cement, which I am very happy for. And the larger projects, more in the cities, we see that. But we also are looking at, you know, different issues in our water system. So all this type of stuff really just helps us and really stirs the thinking and how can we do things better. So I appreciate the collaboration and working with you. So thank you.
You are very welcome. We're a public agency, so everything we do is publicly available. I put our website up there, so feel free to go and you can download all the stuff associated with our stormwater or any of our other research themes, including raw data if you want it. So, all right. Thanks, everybody.
Thank you. Desirae, are you ready?
Never.
Our next presenter is going to be Desirae Braga from the Ventura County Resource Conservation District. She's going to be talking about one of her major projects concerning monarchs and pollinator habitat.
Sweet. Thanks, Heidi. Can you guys see my screen?
Yes.
Sweet. Okay, I'm going to touch a little bit about monarchs and their biology and what we're doing here at the RCD to help their population decline. So monarchs are in the Danid family. There are lots of species. They're found worldwide. Our Western monarch is Danis perplexus, which in Latin literally translates to sleepy transformation because of their reproductive diapause. These guys are effective pollinators, and they're also a flagship species just because of how charismatic they are, as well as a cultural symbol. And they are in population decline. So because they're a flagship species, they're a great symbol for the decline of insects around the world. They also have toxicity to other animals, so that helps them from predation. Just some kind of fun facts. Monarch wings are made of tiny scales. You can kind of see in this photo, these scales give them their color, so it reveals their age when monarchs are kind of less bright once the scales are taken off. Their bright color is a product of aposematism, and this is just like a form of evolution that favors the brightly colored monarchs from predation. They exhibit sexual dimorphism, which I'll get to in the next slide. But I wanted to touch on a recent study from the University of Georgia that studied the monarch wing spots, these white spots on monarch wings. They compare different species of monarchs. They found that Eastern monarchs, the population that goes to Mexico, had slightly less black and more white. And this pattern color could interact with sunlight, white not attracting as much sunlight to create subtle temperature differences on the wing, and that could alter airflow. So this could make butterflies fly more easy as they travel to make that migration. And I'll show you on the next slide kind of a comparison of different Danid species. So the Florida queen is a non-migratory butterfly. And you can see they have less white spots than our migratory monarch. So this is still a new study, but very interesting just to see how they have this success in the long migration. And then, like I said, they exhibit sexual the males are different than females. So females have more black on their veins in the wings. Males are also bigger, and they have these hind spots on their hind wings. And so this is kind of just to show a difference between the Eastern monarch and the Western monarch. As you can see, the Eastern monarch is the one that is more talked about, that makes that long migration from Canada all the way down to Mexico and back up. And our Western monarch is what we have here in Ventura County. They overwinter along the coast, and they have breeding and migratory habitat more inland. And so the Rockies is what divides these two populations. And that's kind of an easy thing to think about when trying to compare these two populations. And then here's just another map from Xerces that shows how the monarchs overwinter on the coast and then they disperse in the spring. Once those warmer temperatures hit, it triggers their migration where they're going to go northward and more inland and seek those milkweeds to breed and lay their eggs. And then I think maybe it's the next slide. This is a Xerces map. So they have all the overwintering sites mapped. And this is a good place to go if you wanted to go see the clustering on overwintering populations. So as you can see on the coast, there are lots of overwintering sites, but there are some inland that just have that good habitat for overwintering. While they're overwintering, they're in reproductive diapause. So they don't move, and that is why a lot of conservationists are seeking habitat restoration, just to have these nectar resources close to the monarchs as they're in this state. So they are clustering in the trees, and we don't want to have milkweed in these overwintering sites or nearby, because that can take them out of that reproductive diapause and make them want to breed and throw off their migration. And in Ventura County, we have 12 known sites. Some of the tree species that are in these sites are blue gum eucalyptus, Monterey cypress, Monterey pine, western sycamore, coastal redwood, and coast live oak. And these overwintering sites provide a specific microclimate condition, which scientists have found has dapple sunlight, high humidity, fresh water, no high winds or freezing temperatures. Of course, this is in an ideal setting and not always true with this habitat destruction that has taken down the populations. So these guys leave their overwintering sites in late March and then reach their northern limit of the range in mid-June. So that's when you're going to see that second migration. Here's just a cool clip just to see the clustering. They look a little bit more active here, probably because it's getting warmer, but normally they're quite still and hard to sight along the leaves of the tree. This is a video from Mexico where the eastern population overwinters. Pretty cool. And then I'll touch a little bit about their host plant milkweed. There's 15 different species in California. The most familiar or the most readily available that you would get in commercial nurseries are Asclepias fascicularis and Asclepias speciosa. But there are a couple of other species, Ariocarpa and Cordifolia, that are less readily available, but still highly prized. These milkweeds are thought to be better because they stay herbaceous a bit longer or earlier in the season. So it's helping their migration earlier on. A good rule of thumb is to not plant milkweed if you are within 5 miles of an overwintering ground. So one thing that I like to tell people is to go on Google Maps and kind of use that measuring tool to map how far you are from the coast or how far you are from an overwintering ground. And if you are within 5 miles or close to 5 miles, I would not plant milkweed, as it can spread parasites and overall hurt the migration of the monarchs that you're trying to help. I would say otherwise to plant native species just as nectar resources. Milkweed also is a great plant for a whole other myriad of invertebrates. There's leaf-feeding beetles, also known as the blue milkweed beetle, seed-feeding bugs, which some people call milkweed seed bugs, and a stem weevil. They also have these bright colors that they get from the milkweed for the toxicity. Tropical milkweed, if you're not familiar, is banned in Ventura County. This is because it retains its leaves in the winter. This can help the protozoic parasite persist throughout the winter. And so when new monarchs come to lay their eggs, they can also be a host of this parasite. And just because it stays herbaceous throughout the winter, it can pose different health problems to the monarchs. So this is banned in Ventura County by the Ag Commissioner. in retail and commercial nurseries and is rated as a B-rated noxious weed. So here's a little bit about some legislation. Monarchs are not listed, so it is hard to get. This hurts us in terms of funding for monarchs. This was listed in the IUCN Red List recently, and the reason being is habitat destruction and climate change hurting this population. But this is more of an educational thing. It doesn't confer any protections or benefits to monarchs. A new thing that came out is pollinator safe plants. And so this is just kind of making sure that your plants are not are organic and don't have any pesticide residue. So they did this study and they found it was over 50% of milkweeds that were, even native milkweeds that were sold in nurseries as organic, had this pesticide residue. So this new labeling can help in terms of being able to safely pick out which pesticide or which milkweed is pesticide free. Another thing I wanted to touch on is captive rearing and captive breeding. These are both done with good intentions. Captive breeding is the practice of keeping monarchs in captivity to create a colony, and then let that colony out in the wild to save monarchs. But this has shown to increase parasites, dilute genetic diversity because it's breeding over and over again and can interfere with research being done, such as tagged monarchs. Captive rearing is when you would take a monarch egg or monarch larva and raise it. But this has shown to reduce population fitness. So in California, it's best to get a scientific collector's permit if you want to raise monarchs. I know I did it a lot when I was young in school. It is a great educational component, but it can be shown to reduce the population fitness and overall migrational success. And so this is OE, the protozoic parasite that I mentioned. Once monarchs emerge from the chrysalis, they can be deformed. So OE has a long name, Ophriocistus electrocicera, and it is present in all Western population of monarchs. And it lowers the migrational success. So this lays on the milkweed itself, and once the mother lays her egg underneath the leaf, the egg then induces the spores. So the larva can ingest it once it emerges, and then it follows the same life cycle as the monarch. So if the monarch makes it to the chrysalis, then it'll emerge deformed, and sadly, they only live for a couple days. So here's some recent numbers from the volunteer population counts. One of them happens just next week in Thanksgiving, and then another one happens New Year's, if anyone's interested. It's the Great Citizen Scientist report, and so it shows since 1970, which we do have less data from, 99% of monarchs have declined from our Western population. And in 2020, the monarch population was at an all-time low. Last year, there was a 58% seasonal decrease. This was just because of weather patterns. If any of you remember those storms we had, it kind of triggered them to leave earlier before we had our counts. But as you can see, the population does fluctuate, but there is an overall decline in monarchs. So this is raising concerns and why we need legislation so we can get this funding to help monarchs. So a little bit about what the RCD is doing. We have a Harmony project, which is a monarch and pollinator flyaway path. So we are doing habitat restorations in all these sites you see here. It's a pretty big project. I think there's like 17 sites in total. We're looking for funding for next year to kind of replicate this. We have overwintering sites as well as breeding and migratory habitat. So some of these inland sites have milkweed, which we will be monitoring later on once these get implemented. And then sites like Big Sycamore and La Jolla, these are overwintering sites that we have a habitat management plan for. And so some of these sites are just planning. Harbor Boulevard, Douglas Family Preserve. These are planning for these plans to be developed, and then we will go for implementation funding next year. And these more inland habitats are just to kind of help give the monarchs some nectar along their path as they're migrating. So what can you do to help the monarchs? We can plant native plants, which is what we're doing at these inland sites. And these nectar resources are great in helping the migration of the monarchs, more as like gas stations or pit stops along their route. So a little bit more about this Harmony project. It's funded by the Wildlife Conservation Board and the U.S. Fish and Wildlife. We rely heavily on volunteers for plantings, maintenance, site prep. There's many different partners in this. And once all these sites are implemented, which I think we have about half right now, we have a larva monitoring protocol. And so I'm hoping to make this into a citizen scientist project. A little bit of education and outreach. We have the Monarch and Pollinator Regional Advisory Committee. We meet I think we meet 2 times a year now. I just reduced it. And then we also have a Monarch Alert, which is the citizen scientist protocol. If you sign up, you get email push notifications that shows you when the monarchs are migrating, what you can do, what BMPs to implement. So this is kind of a great resource for both farmers and ranchers or just your everyday gardener if you want to learn how to help. You could also become a volunteer. So if you go on our website, www.vcrcd.org, you can sign up to help with our plantings, MRAC, or any of the other great things we have going on here at the RCD. Okay, I know I kind of sped through that. Do I have any questions?
No questions right now, but I might have a couple questions for you, Des. Are you, like, let's say someone works for the city or something, and how big a land are you looking for habitat restoration for monarchs? Like, can it just be like a little plot, or does it have to be like a certain size to be effective?
There's no requirement. So whatever you got, we'll take it. I think we have 3 sites picked out so far for our next round of funding. Obviously, the bigger the site, the more habitat that we can provide.
Go ahead, EJ.
Yeah, thank you. Very interesting presentation. On your map, you showed the breeding areas along the coast, and then you showed arrows going inland, and they look like they were to the non-breeding areas on the coast and the arrows to the breeding areas. It looks like they went way inland. At what point do the breeding areas start, I guess, is my question.
So all of the breeding areas is what you see. That's not on the overwintering, not on the coast or listed on the Xerces site for overwintering habitat. Okay. So all of the overwintering sites are designated on the Xerces site, and they come back each year to these same sites. Typically within 5 miles of the coast, but there are a couple that are more inland.
So would you get breeding areas in like Simi Valley or Santa Clarita, or if they had the right habitat.
Yes, yeah, exactly. If you are within 5 miles or outside of 5 miles of the coast, that's great breeding habitat.
Okay, Brooks?
Desirae, what is the I guess ideal spacing as far as distance goes between nectar sites? When you mentioned you mentioned the habitat site could be pretty small. What do you think the ideal distance would be?
The goal of the Harmony was just to kind of connect these wildlife corridor or spaces in between these wildlife corridors where there wouldn't be good habitat for these nectar sites. So there is no like rule for how close these nectar sites can be. But what I am looking for this next round of funding is continuing following on that migrational path, the flyaway path as they call it, and just kind of connecting those sites in between where we have high quality habitat and wildlife corridors. And I could go back to that map to show you too.
Okay.
Yeah, these guys right here. So yeah, just kind of looking for what's in between these wildlife corridors to connect high quality habitat.
All right, thanks.
Thank you. And Chris?
Hi, can you hear me?
Yes.
Okay. This is Chris Ament from Native Monarchs, and I'm working with Desirae on the Sterling Hills golf course habitat. Desirae, I wanted to kind of get your take on what the RCD thinks about the endangered reversal that hit the media a couple weeks ago. So the IUCN last year had hit all the news media outlets, basically putting the monarch butterfly on their endangered list, which was their red list, although it didn't have any federal jurisdiction like Fish and Wildlife does. However, they just reversed it, put it out in the media that they didn't have the correct population studies. So it's causing a giant wave of confusion for people that are saying, you know, is it endangered? Is it not endangered? Is it at risk? Is it warranted? My fear is that everybody kind of says, oh, well, it's okay now, and we don't have to do projects like this. So, you know, as far as you and I go, you know, we push forward with these habitats and still plant native milkweed and get the awareness out. But I think it's good to know, at least for maybe other people that aren't in the know of what's going on, and that message is out there. So as part of even working with Brooks and NRCS and the pollinator habitat programs, these are really, really important programs that I worry is getting kind of overshadowed by these kind of like media headlines that aren't really accurate.
Yeah, I think it's a shame that they did that reversal. And we're all confused. I don't know if part of it was maybe they got the populations mixed up and they were trying to list the eastern population, which is doing slightly better than the western population. But yeah, even though it has no legislative backing, it's still a bummer that this education kind of it makes people say, oh, okay, well, this monarch is doing fine. I don't need to implement any of these BMPs that people are recommending.
Yeah, I agree, and I worry about that.
Yeah.
Good questions. Coming from the public side, we have a lot of great outreach, and as a parent, we have milkweed in at my house because the teacher said, hey, you guys should have this and let's grow. But I appreciate really having a more focal point, but for our community to be educated as well, so that we're not just planting all these different things. Are we looking at key locations where we can educate, utilizing the museums, utilizing Channel Islands, CSU Channel Islands, the Oxnard College, Ventura College, those locations? You know, you have Tierra Rajada is part of the wildlife corridor. We have Underwood Farms. Those are places where we could draw community and education on there, similar to, say, for example, Santa Barbara Museum of Santa Barbara. They have that beautiful Monarch display every year. Are we looking at creative ways in educating our community and building these sites so it's a win-win? And then the other question I have is with our agriculture team, Corinne Bell and so forth, if we give parameters as to what we're looking for, how many plants, what needs to be done, we have so many ranchers that they don't need a wildlife corridor. They have wildlife at their farms always. And I think that people don't think that sometimes, and it really frustrates me because wildlife is one and the same, and they're all environmentals too. So how do we get the word out in that way? Because I know Ventura County steps up like you wouldn't believe. So what do we need to do? How do we help you? I guess is my question from a standpoint of I do a lot of communication, so I'm trying to understand.
Yeah, I think outreach is a really big tool. And so using things like this Monarch Alert program that we're trying to develop and getting that outreach out to farmers and ranchers who don't really think that they need to have this habitat on their land or it's taking up space, or yeah, it's good to get the word out. And so I know
I don't think it's that they don't want it there. What I'm saying is that they would if they knew that this is what they needed,
Right.
support. So don't think that they're negative in any way, shape, or form.
No, right.
They're always looking at opportunities.
Mm-hmm. Yeah, and so just getting the word out on how they can help, what BMPs to implement on their land is great. And so we have a couple initiatives at the RCD that we send email push notifications. We have a monthly newsletter that goes out. And so just helping in that way of outreach would be great.
Okay.
And
then the college
to add,
I would say.
yeah, if I could just add, part of our grant also funded the creation of the Monarch Alert program, which, as Des mentioned, it's meant for the just normal households throughout Ventura County, but also for farmers, like anyone who does want to know what they can do to help during different times of the year. It does give seasonal updates of like, this is when the migration should be starting. Look into alternatives to your normal pesticide use and like that kind of stuff. So I'll include the link to that sign up and maybe some of just our marketing for that so that other people can share it in their newsletters and things like that. Peter.
Yeah, thanks for the presentation. Maybe I missed it, and I know there's discussion more about outreach and education, but are you reaching adequate public? Are you reaching the public adequately? And if you need assistance with reaching the public adequately, are you reaching out to public agencies or partners, private partners about cross-posting on, Facebook, Instagram, all the other media channels and media sources? Because I do think hey, the city of Ventura, are you in communication with our communications team and the PIO? We're happy to do some most likely cross-posting, cross-pollination with you to get that message out. And I think that a lot of the other organizations on this call may also be able to support the messaging if needed, because we've got bigger platforms that we could potentially utilize and we could have some benefits.
Yeah, that'd be great. I think it ends up we keep outreaching to the same people on our listserv each time. So definitely doing that cross-pollination and posting these things on different sites would be great, because a lot of this is reliant on volunteer support for all of our restorations. And we're kind of getting them all online at once. So it's a little bit overwhelming to do all this outreach. So yeah, we would appreciate that.
Yeah, just send me an email. So if you instigate with me, I can push you over to the comms team at the city.
Okay.
Or you could just blanket email everyone that says they have communication expertise and they can kind of plug you into their orgs. But yeah, send me an email. I'm happy to try to help you out.
Sweet. Thanks, Peter. Chris, you had your hand raised.
Oh, yeah, I just wanted to follow up with Kelly's question about outreach. So we have a program right now that's going on at CSU Channel Islands. So we have, if you're familiar with the campus, there's a big MODOC garden that we have there that is where the students can go and raise vegetables and fruits. But we started last year installing a Monarch and Pollinator garden in the back. So this was part of a biology class where, in lieu of writing a research paper, they could take a plot of land and they could plant a monarch and pollinator habitat in approximately a 10 by 10 space. This started as a project. I was working with Dr. Alarcon in the biology department that has just turned into something so awesome. I can't describe it. You have to see it. But all these little plots that have basically started to grow together. There are so many butterflies and pollinators out there that they're going to start to, we want to make it a permanent habitat, keep adding to it, where semester after semester, students are invited to take over a plot, maintain a plot, add a new one. But this is, I think this is the start, because Channel Islands is starting to realize that community service events coming into the garden will be really important going forward. We're trying them now. We've done a couple of them. We're going to do a harvest festival on Halloween. But bringing in the public, the Boy Scouts, the Girl Scouts, doing weeding and planting sessions, and just basically seeing this habitat. We really want to develop this garden. And I think this could be your template for all other schools that have a little bit larger pieces of property, where you make a pathway through there that they stay out of the habitat, but they can still view the habitat. But then you can also get projects like student monitoring and data collection. And in the West, we are really low on the Western monarch data collection. Everything is pretty much done in the East a lot, but we definitely need more. So that is something, that anybody wants to come out. You can always, I'll put my contact information in the chat. You can always come out for a tour. I definitely think it's worth seeing it right now, because it looks like spring. Everything is blooming, and there's so much activity out there that there are butterflies I've never seen before in my life. I have to get my book out. So as far as that goes, I think that's the template. We want to try to do that in more places.
I appreciate that, because I spent the day with Dr. Yao, and we were in Santa Paula High School, and we were with the Boys and Girls Club, and there's so many ways in which you can educate the community and build larger areas so that people can see it, enjoy it, and get more information. And I think the education side is really an important part. So thank you.
I agree. Thanks.
Okay, thank you. It's time for us to move on to the next presentation, but Desirae just dropped her contact information, and we'll be following up with Peter about outreach for that. Next, we have Brianne Sakata to talk about the Lake Castaic algae bloom that occurred earlier this year, and just give us some information and updates about that. Are you ready?
Are you seeing my presentation?
Yes.
Okay. And it's the notes view?
No, it looks normal for us.
Great. Well, thank you, everyone. Oh, are you hearing me okay? It says my Zoom just crashed. I don't know. That was weird.
I can hear you and see you.
Okay, great. Okay.
Oh, no. You're muted.
Did that unmute me?
Yeah, you're good now.
Okay, great. Sorry about the technical difficulties. Thanks everyone for having me here today to talk about the algal bloom that's happening at Castaic Lake. I'm Brianne Sakata. I am a senior environmental scientist with the California Department of Water Resources, and I'm the lead for the State Water Project's Harmful Algal Bloom Monitoring Program. And the photo you see here on my slide was taken at Castaic Lake a
Oh,
couple
we
of...
can't see your slide anymore.
Oh no, man. Okay. I think I might have to reshare. I think that's why Zoom told me it crashed.
Yeah.
Okay. Sorry about this. I cannot even get Zoom to open right now to be able to share my screen. I might have to leave and come back in to the meeting. Yeah, okay. I will be right back.
Okay. Dawn, you might have to go in front of her if she's it's having some tech issues she needs to work
Okay,
out.
hopefully I can jump. If you want me to just jump in. Or...
She just joined, so let's see how she does, and then we'll decide.
Okay, can you hear me okay?
We can hear you, yep.
So sorry about that. I'll try to reshare my screen. Maybe I'll leave my camera off in case that's what crashed everything before.
Yeah.
Okay.
We can see it.
Oh, great. Okay, perfect. All right. Okay, so like I said before, I think you guys could hear me. You couldn't see my slide though. I'm Brianne Sakata with the Department of Water Resources, and I'm here to talk about the harmful algal bloom at Castaic. And the photo there was taken at the lake a couple of weeks ago. So in this presentation, I'm planning to go over just some basic information about harmful algal blooms and their impacts, and the state guidelines for recreation and drinking water. I'll talk about our algal bloom monitoring and present the information we currently know about this harmful algal bloom and how it relates to the watershed, followed by some next steps and some links to some resources. So harmful algal blooms, also known as HABs, are most often caused by cyanobacteria in freshwaters. Cyanobacteria are also commonly called blue-green algae, and they're a naturally occurring part of the ecosystem, and just the fact that they're there doesn't always mean there's a problem. However, under ideal conditions, they can grow rapidly, causing an algal bloom. Ideal bloom conditions can include warm water temperature, calm or low flow conditions, and high nutrient levels. Cyanobacteria can produce toxins called cyanotoxins that can be harmful to humans and animals. So some signs that a harmful algal bloom is present can be scum, foam, or mats on the water surface that can wash up on shore. They can have a streaky appearance like what you saw in the previous few slides with the photos of Castaic. They can also look like spilled paint or look like small green particles in the water. And the colors can vary from green to blue-green, brown, or white. And these three photos on the slide here, they're not from Castaic. These are from other State Water Project lakes that I included just so you can see some of the color variation we see in blooms. Bloom conditions can change rapidly, and wind or waves can move and concentrate a bloom around into different regions of a lake. So there are a few ways that people and animals can be exposed to the toxins produced by harmful algal blooms. The main way is through water recreation. People may accidentally swallow water, they'll have skin contact with the water, and then activities that can create spray like water skiing or jet skiing can cause the toxins to form aerosols that can be inhaled. Exposure can also occur through eating fish or shellfish that were caught in a water body impacted by a HAB. And drinking water is also a possible exposure source. However, the water treatment process does normally remove cyanotoxins. In people, exposure to cyanobacteria or cyanotoxins can cause eye, nose, mouth, or throat irritation, headache, cold and flu-like symptoms, rashes, or gastrointestinal upset. Animals, and especially dogs, are more susceptible due to their small body size, and their exposure level tends to be higher because they'll drink the water, they'll lick algae off their fur, and they may eat the algal mats that wash up on the shore. In animals, cyanotoxin exposure can cause diarrhea, vomiting, convulsions, and even death. So California has set some recreational guidelines, and they're for 3 different cyanotoxins: microcystins, anatoxin A, and cylindrospermopsin. And there are 3 different advisory levels: caution, warning, and danger. The Castaic Lake algal bloom is producing microcystins. So the caution trigger for microcystins is at 0.8 micrograms per liter. The warning trigger is at 6, and the danger trigger is at 20. And Castaic Lake is currently under a danger recreational advisory. And these are guidelines that are voluntary to follow. They're not regulations. And these are the 3 advisory signs that list the recommended precautions for each advisory level that should keep people safe during recreation. At the caution advisory level, swimming is still okay, but people are cautioned to stay away from algae if they see it in the water or on the shore. At the warning and danger advisory levels, swimming is not recommended. The danger advisory that's currently in place at Castaic Lake means that people and animals should stay out of the water. Fish or shellfish caught in the lake should not be eaten, and raw lake water should not be used for drinking or cooking, even if it has been boiled or filtered. These are guidelines for cyanotoxins in drinking water. The EPA set 10-day health advisory levels for microcystins and cylindrospermopsin, and the values shown there in the top table are the amounts that could be consumed daily in drinking water for 10 days without adverse health effects. For microcystins, which is the toxin present in Castaic, the advisory level is 0.3 micrograms per liter for young children and 1.6 micrograms per liter for older children and adults. The California notification levels for cyanotoxins in drinking water for 4 cyanotoxins are shown in the bottom table. These are levels set by the state that if they are exceeded in drinking water utilities are supposed to inform their customers. You can see that there are acute notification levels that are for a 1-day exposure, and short-term notification levels that vary in length from 1 month to up to 3 months. For microcystins, the acute notification level is 3 micrograms per liter, and the short-term notification level is 0.03 micrograms per liter for up to 3 months. So some information about where the water in Castaic Lake comes from. Most of the water in the lake is from the State Water Project, which begins in Northern California at Lake Oroville, and water is moved south through the state through the California Aqueduct. And this map is showing the State Water Project's West Branch, which includes Pyramid Lake, Elderberry Forebay, Castaic Lake, which is also known as Upper Castaic Lake, and then Castaic Lagoon, which is also referred to as the Lower Castaic Lake. So water in Pyramid Lake exits and moves through the Angeles Tunnel, where it flows through Castaic Power Plant and into Elderberry Forebay. From Elderberry Forebay, water can be pumped back into Pyramid Lake or can be released into Castaic Lake through the stream release that is near Elderberry Forebay Dam. And once water has moved out of Elderberry Forebay into Castaic Lake, it can't be pumped back to Pyramid Lake. Castaic Lake does also receive a small amount of natural inflow from Elizabeth Canyon Creek and Necktie Creek. DWR routinely monitors the State Water Project for cyanobacteria and cyanotoxins, including at Castaic Lake and Castaic Lagoon and upstream of Castaic Lake in Pyramid. Our monitoring season generally runs from April through October, and we monitor at 2 different types of locations: water quality sites and recreation sites. Water quality sites are locations where water is delivered to contractors, and on the map there, the red star is marking our water quality sample location at the Castaic Outlet Tower. Water quality sites are usually sampled twice a month. Recreation sites are marked in the blue stars on the map. Those are generally at swim beaches or boat launch locations. And the blue stars on the map are marking our recreation sites at the Castaic Lake main boat launch and Castaic Lagoon swim beach. Recreation sites are sampled once a week from Memorial Day through Labor Day, which is the peak recreation season. If toxins are detected at any of the sites, if they're not already on a weekly sampling frequency, we do increase the frequency to weekly. And at the end of the season, if toxins are still being detected, we continue to monitor until they're no longer detected. So the samples that we collect are sent to our contracted laboratory, and they're first looked at under the microscope to see if there's any potentially toxigenic cyanobacteria present. And the lab, based on what they see under the microscope, they will either do toxin analysis on the sample, or they'll say it's not recommended. And they'll also determine which toxins to analyze for based on what they see and based on the site history. We share our lab analysis results with recreation managers, the State Water Board, water agencies, public health departments, and many other interested parties. DWR recommends recreational health advisories to recreation managers based on our monitoring results. So if one of those trigger levels is exceeded, we immediately notify the recreation manager, and then the recreation manager decides how they want to implement the advisory. For example, if just one location exceeded a trigger level, the recreation manager might decide to only post an advisory at that certain area, or they may decide to post the advisory for the entire lake. So this graph is showing the microcystin concentrations at Castaic Lake over the 2023 monitoring season. And as you can see, we have one very high value that's 464 micrograms per liter, which is what triggered the lake closure. And that high value makes it so you can't see any of the other details in the graph. So here's the graph with a break in the scale on the y-axis, so you can now see the details. That yellow horizontal dashed line at the bottom is marking the caution advisory level of 0.8 micrograms per liter. The orange horizontal line is marking the warning level, and then the red line is marking the danger level. And the 3 sites that we sample in Castaic Lake are graphed on here. The boat launch is in blue, the outlet 1 meter depth is orange, and the outlet surface is in green. And you can see there was a caution advisory triggered by the sample collected from the boat launch on August 28th. Then the danger advisory was first triggered by a sample collected on September 18th at the outlet 1 meter depth. And then the sample from the outlet surface collected on October 9th was that very high value of 464 micrograms per liter that caused LA County Parks and Rec to decide to close the lake to boating. I'd also like to point out, you can see, I mentioned that bloom conditions can vary. You can see how varying the toxin levels have been since that danger advisory was first triggered. They're down the next week, up the next week, and then back down. So that's why we typically leave an advisory in place until toxin levels have remained low for 2 weeks. So here's what the species that are present in this bloom look like under the microscope. There are 3 species of cyanobacteria in the bloom that are capable of producing microcystins. We have Microcystis on the left, Dolichospermum in the center, and Warneckinia on the right. And then there's a Phanizomenon in this bloom also. That's the micrograph at the bottom. That one is not a microcystin producer, but it can produce other cyanotoxins. And in that sample that triggered the lake closure, Microcystis was the dominant species. We also have the ability to look at the bloom with satellite imagery using a tool that was developed by the San Francisco Estuary Institute and the State Water Board. So the cyanobacteria index scale is shown on the left. It's black means cyanobacteria are absent, and then the cyanobacteria abundance increases as you move up the scale from blue to red. So you can see in the image on the left, the bloom started in Elderberry Forebay, and then it moved down into the lake, spread throughout the lake, and then intensified. So the third image from the left was taken on October 9th, and that was the day that the sample was collected that triggered the lake closure. And here's what the current conditions look like. This was the satellite image. I just pulled this off the website this morning. So it's the 10-day average as of yesterday. And the graph there is showing that cyano index over the past month. And you can see, according to the graph, the bloom is leveled off and it's remained stable for the past couple of weeks. So there's understandably been a lot of concern about what this bloom might mean to water users and the watershed. Castaic Lake is a drinking water source. However, we don't believe this bloom is impacting the source drinking water. Water that's delivered through the outlet tower is drawn from deep in the lake below that surface layer that's currently being affected by the bloom. If there did happen to be cyanotoxins in the source water, those can be removed in the water treatment process. We've also been hearing from a lot of people who are wondering if this bloom in Castaic Lake is being moved downstream. Water is not currently being released from Castaic Lake. The last time it was released was on October 6th, and that water was also taken from deep in the lake below that surface layer that's being impacted by the bloom. Water from Castaic Lagoon can be released to Castaic Creek, which is a tributary to the Santa Clara River. The creek normally only connects to the river during wet winter periods. And water is not currently being released from the lagoon into the creek. The last time it was released was on October 9th. As for recreational users, once the lake reopens to recreation, just follow those recreational advisory guidelines on any signs that are posted around the lake, and those are designed to keep people safe during recreation. So some next steps, we will continue to collect samples weekly in Castaic Lake to monitor the toxin levels. And then any recreational health advisory changes will be made in consultation with LA County Department of Parks and Recreation based on our monitoring results and the visual bloom observations at the lake. Any advisory changes will be posted on DWR social media and the DWR Algal Bloom website. So here's some resources. Our Algal Bloom website that I just mentioned is linked at the top. That website has the status of all of the state water project lakes. And then there's a link there also to the page for Castaic Lake. The California Harmful Algal Blooms portal is a good resource. That website shows blooms that have been reported all throughout the state. And there's also a link on that page where you can report a bloom if you see one somewhere. And then that satellite tool that I mentioned is linked here. Anyone can use it, and you can see satellite imagery for all the lakes in the state, as long as they're big enough to be seen by the satellite. It doesn't really see really small lakes. Okay, with that, I'm happy to take any questions.
Thank you. I really like those resources you included at the end.
Thank you.
Those are super useful. Does anyone, I have a question, but I'm gonna see if anyone in the audience has a question first. Oh, someone is asking what may have caused this bloom.
Well, that's a good question. I can't really pinpoint it to one certain cause. There are conditions that can favor blooms, but I can't point to any one thing saying, oh, there were high nutrient levels and that's what caused the bloom.
EJ.
Yeah, thank you. Do you know what the satellite is looking at to identify the algal bloom?
It's looking at phycocyanin, which is a pigment in found in cyanobacteria. The satellite, actually, they just launched it now. You can look at chlorophyll A concentrations on the satellite also.
That's amazing. Thanks.
Just as a question, we get bloom like this in many different lakes. A lot of the triggers from my background, from what I understand, is that it's also components of the heat, the temperature of the air, the temperature of the water, all of that. Are we tracking that along with your water quality on your days? Or what the water level is? If it's lower on the water scale, lower lake level versus higher, more concentration. Do we look at that in any of the data?
We do have that data. I don't have it all pulled together in one place, but we are taking different water quality measurements at Castaic, like water temperature. And yeah, that is data that we have available, but I don't have it all pulled together in one place.
Okay, because that's also an interesting one too, in regards to what do you do to counter that bacteria? Is there anything that you can do to counter it? And does it hurt any of the other environment around the lake because of the bloom?
We have another question in the chat saying, are there contributing factors in human use that residents can be aware of to reduce the risk to waters?
Sorry, can you repeat that again?
Yeah, just are there contributing factors that human use, like contributing factors that we should be cutting back on or being more aware of that can help prevent these types of blooms?
Well, I can't speak specifically about Castaic Lake, but I mean, nutrient inputs can trigger algal blooms. So, use of nutrients like fertilizers within a watershed could possibly contribute nutrients to a water body. I don't think that's the case at Castaic Lake. But yeah, that is definitely a good practice, is to reduce nutrient inputs.
And then I had a question. You had that thing for like reporting algae blooms. If I'm like just going on a hike along a river and I see like kind of more algae than I would expect, is that something people report? Or is it just for like standing water?
They report it for all types of water bodies. Yeah. So yeah, if you go to Maybe I should go back to the resources page. Yeah, that mywaterquality.ca.gov/HABS website. If you click on the report a bloom link, they'll ask you for all kinds of information. They'll ask you for photos, maybe GPS coordinates of the location. But yeah, you can report a bloom for any type of water body, lake, stream, river, anything. And then someone from the water board usually will reach out to you for more information. And then the information reported through their portal, once they verify it, goes on to a map. So there's a map on that portal that'll show all the bloom reports at water bodies in the state, and they'll put your bloom report on the map. So if you want to go somewhere and are wondering what the conditions might be like, it's a good map to refer to just to see if anything has been reported. It doesn't mean there isn't anything there, it just means that no one's reported a bloom for your water body.
Cool. Any last questions? One last, are there mitigations that happen to reduce the severity or length of the bloom?
For a bloom this size, really the only mitigation would be an algicide treatment. We don't currently have plans to do that.
It's just good. It's good to know that, or to at least communicate that. And then also communicating to the residents that if you do touch it, wash with soap and water, that kind of stuff, so that we're not transferring contaminants.
All right. Thank you so much for your presentation. Would you be open to sharing those files with me so I can post it when I post the recording later?
I think that would be okay. I need to check with our public affairs just to confirm it's okay for me to share all the information on there.
Okay.
But I'll be happy to if
At the
I'm
very
able to.
least, just like the links that you have on here.
The links, sure. Yeah, definitely.
Cool. Thank you so much. Next, we are going to be learning about invasive species from John Beall. Are you ready to go, John?
Yeah. Okay, so go ahead and I'll share. Let's see. Okay, and then you want to go to start. Right, that, is that right? What? Is that? Oh, that go? Okay. And then where's my is it on? Let's see. The my sound. All right. Hello? Can you hear me?
Yes, we can hear you.
Okay. All right. Yeah, I'll try to go pretty quickly because we're running a little late. And then okay, so we use iNaturalist for the ag department for checking, finding out if there's any new weeds, basically invasive species. And one of the, I'll kind of go through this outline for the presentation. Normally it's Taylor Wallace, my coworker, is doing this, but she's at a conference, so I'm working on it. Okay, so basically we're gonna go into the talk a little bit about the ecology and early detection of invasive species, how you can help. Citizen science is iNaturalist, and also a few key invasive species that we're looking at in Ventura County. Anyway, so we have a lot of ecosystems in Ventura County, obviously, and we have different weeds in each of them. One of the things is that we do have this amazing group of native plants, and one of the things is just stopping to look at what we have, our ranches, our open space and creeks, and we can do very simple things to protect and restore them. So one of the things I want to get into is the terminologies. So native is a species that evolved in an area from a long, very long time, a deep time context, millions of years. Non-native, a species that arrived in an area that did not evolve in the system. And invasive that can, not all non-natives are invasive. There's a lot of plants that are out there that are not native, but they're not aggressively taking over particular areas. And anyway, so some of the invasive species are a major threat to biodiversity. This over here on the left, you see stinknet, and that is in Arizona around Phoenix. It's taken over quite a bit of in certain areas and just completely covered it. We have Tamaris, Cape Ivy in some places along the coast. We have it over at Foster Park. One of the things, this is just a recent report from the UN, and we had somebody from this group speak at our weed management area. 60% of the global species extinction have been caused solely or alongside other drivers by invasive species. So it's a huge amount of money. We're in 423 billion worldwide annually, and so it's a lot of money. One of the things that in our department, we focus a lot on this chart on the left, where we're dealing with introductions and early detections. And one of the problems is, how do you find out when something comes in? How do you do detections? A lot of historically ag departments would go out and drive around and look for weeds along roads or the like. And so anyway, at lower levels, it's more possible to eradicate some of these things. Like, I mean, when you talk about eradication, it's like completely removing them, or getting pretty close. And there has been some eradications, but they have to be. You have to have early detection. You have to really go after it in a strong way, and then later, some of the other weeds. A lot of times, by the time land managers begin to and public awareness happens, some of these things are out of control already. But that doesn't mean that you don't have to do anything about them. Our focus in our department a lot of times is over in the left side. In our weed management area, we are dealing with the entire range of weeds, early detection. We have presentations and things about how people can deal with, different, more common weeds that may be impossible to eradicate, but need to be dealt with. So how can you help ID citizen science and iNaturalist? So anyway, citizen science has a long history, and over on the right is very interesting. When I was helping to clean up our fumitorium, I weirdly found this box of books, notes, and it was this thing on the right where this person started taking information about climate, temperatures, and this particular person, this one over here on the right, was done up in upstate New York in 1859, and the group of farmers that were given thermometers and rain gauges and such were the initial citizen scientists that started the National Weather Service. So we've done this for a long time. It's not just really recent. And in more recent times, there's a lot of people want to get involved, and a lot of it is data collection. So anyway, so what is iNaturalist? It's a application that can be used to you can identify plants and animals, and then it will also place these on a map and in iNaturalist. And one of the things that we're finding is the middle one is the spotted lanternfly, which is a huge problem in the east. In our department, we don't have it established in California. It's a very voracious pest, but there's not a lot of times these new insects and such come in And they're normally the way that CDFA and our department kind of would deal with it is by trapping, but sometimes you can't put together a lure or a trapping program that quickly. So iNaturalist can help out with that. So that's why. So there's a lot of ways to engage with iNaturalist. One of the things that main thing is adding your own observations. And the thing about it, it's super easy. It's a very easy application. And it's sort of like if you want to go out with your family and just check out things, it'll help you give you an idea of what you're looking at. So anyway, one of the things that we do is we work on confirming IDs. There's a lot of iNaturalist, and we have a good group here today. That iNaturalist is a big net that is got a lot of flaws. People are putting in observations that may be off a bit, but there's the volume of them is so great. There's like 162 million observations, and there's probably in Ventura County, I think we saw for plants, there was like 15,000 observations in plants so some of those are invasive plants that we're interested in. So anyway, adding your observation via smartphone, you observe with a camera, the app is right here, iNaturalist. You basically just download it. It's very simple to get into. You observe and you take a photo of maybe 4 or more pictures of the plant or in case an insect. But the main thing is that with a plant, if you can take flowers, seeds, features of the plant that will be helpful and make sure that they're clear. And then you upload the observation. Take multiple photos from different angles, different plant parts, maybe take a single plant picture. Sometimes if you have a plant that's mixed in with a bunch of other plants, it kind of makes it more difficult for iNaturalist to give you an idea of an option of what it is. Because basically, based on these photos, it'll give you an option of what it might be, and sometimes they're wrong. So the better photos you have, the better chance you have of getting a correct observation. A lot of times, what we will do is stay at the genus level if you're not sure what it is. And then the other thing is that after you take your iNaturalist location or your shot and you make your ID and your observation, other people will agree with it, and they will often make ID corrections. So it's basically no hard feelings if anybody don't feel like you're messing up. Before you use the features, you need to set up an account and sign in. And then the desktop looks like this, and you can see your observations. You can edit them and correct them. And a lot of times, you'll add an observation and somebody else will agree with it. And let's see. So then one of the things is that under community, there's projects. This is a very useful tool for different people. So it tracks a lot of data, date and time, location, species, information. Sometimes for birds, if you're a bird person, you can get sounds and it will help you decide what it is. So for our department, some iNaturalist observations turn out to be new and uncommon invasive plants. iNaturalist can act like it's like a net, a big net, a net that sometimes has some errors in it, but it helps us capture these important finds, because otherwise we would have to drive all over the county. There's places where people are going in the Los Padres that we're never going to go. So we verify these observations online, and sometimes out in the field we go out. If it's a very important rare weed, then we move the observations to CalFlora, which is an application that is more standard for for our department and a lot of different groups. And these observations help us, when we have these observations, they help us prioritize which ones are more likely to be possible for control or eradication. So the projects feature in iNaturalist is really a useful one. What you can do is, as we've done in Ventura and Los Angeles, we work with Los Angeles, and we have like a group, a combo of these two entities that are part of the South Coast Early Detection Rapid Response. And our project will capture all of the iNaturalist finds that are rare or A or B rated weeds. And that provides us with a list that we can check out and see, okay, well, maybe that's not really what it is, or yes, it is, and let's go find out, or maybe we need to check it out, go find out where it is. So one of the things we work with county parks, we've been outreaching to different entities, and that's been a big part of what we're doing lately, is presenting information about the weeds in our county and iNaturalists, because if we can get particular groups or entities that are out there looking at plants, and they're more likely to be the ones that could find some of these rare or rare invasive A-rated weeds. So, but one of the things that you can do if you have a project, you can have a list for a park, and that's one of the things we just had a training with county parks a couple of weeks ago. So a project, you can hover over to make it, hover over the community, click projects, start a project, and this is our project, the list that we have for Ventura and Los Angeles counties. And these are kind of rare weeds that we don't have that we're looking for. So if any of these, essentially, even if somebody that doesn't know anything about us or is randomly, a random person is out there in the middle of nowhere and he clicks on and finds one of these, we'll know that, hey, there's some Scotch thistle and it's way up here, let's check it out. Or spotted knapweed, or these are things that are rare that we think that we might be able to eradicate. So anyway, so a BioBlitz is something that often happens with iNaturalists, and what it is if you want to try, you have a park, let's say a county park or an entity like Ventura Land Trust. We talked to them a little bit about this. They're one of our partners in the weed management area, and they may do this, is you pick a time and day, you create a project in iNaturalist. So it would, in this case, it would be something that would be everything that's found in a particular park or public land. And then you advertise, you invite the community to join, you get people to come on a day, and then they go out and they just basically it's anarchy. I mean, that's one of the things about this. It's not anything that that has to be super organized, but you basically get people out, they come over and they come to the park, and they with their phones and their iNaturalist app, and they start looking for whatever they see and start taking observations. So anyway, this is there's training materials, iNaturalist. If you're targeting specific species, you can develop training materials to share, and you can have walks. Sometimes you usually want, if you're going to do a BioBlitz, you maybe want to have somebody that's an expert in native plants or weeds or something at that, or insects or birds, and you have them come to the BioBlitz and join in, so that you can have information in that way. So anyway, there's a lot of information with iNaturalist.org and different guides, and also Cal Academy, which is one of the entities that started iNaturalist, has a toolkit. So I'll quickly go into key invasives in our county and sort of a little bit about them. So again, we go after, in our Ventura County, we work agriculture department. We're after more what they call A and Q rated weeds that are rare but very invasive, and we go after those. And then we also, with our weed management area, we go after all the weeds and we provide information and different people that speak to how to deal with it. Like we had somebody talking about grazing and mowing at parks for grasses, that invasive grasses that are not going to be eradicated, but they need to be dealt with. So anyway, so here's some of the kind of rarer weeds. This is Pancrantium meridimum, and it's over right at in Buenaventura Beach, right near on Harbor and near Vista Del Mar Road. And it is very beautiful. In fact, in the middle, you see that it's from Crete and the Mediterranean. And we've talked to people over in Italy about this, and it's rare over there. It's a rare plant, sort of, but it has been moving in along the beaches. And so this spot is one of the few in all of North America. It's not native, and it's in Europe, it's moving out of the Mediterranean all the way up into France and England. So we're concerned about it in our area, even though it's incredibly beautiful. So spotted knapweed, you can see Pine Mountain Club. There's 2 locations in all of Ventura County that we found, and one of them we did find from iNaturalist. Somebody randomly found one on the top of Rays Peak. We went out there. There was about 15 plants. For the last 3 years, we've been going out there, and finally there was like 1 plant. So that's the power of how iNaturalist can really help with us, because there's no way that we would be out there on the top of Rays Peak and find this particular plant. There's just too many acres of land in Ventura County. Skeleton weed is another one. We have it over in Palo Camado Trail, and it's the only location in all of Ventura County. And again, we're looking for somebody that might find another one of these. There's quite a bit in Los Angeles County, but in our county so far, there's only one. But stinknet, and this one is, I know that there is some in the Santa Clara River, but it is, again, I showed you an earlier picture that it really is the big thing about this is you can see it when it dries out. It's a very huge, it can grow like 3 inches or 4 feet tall. And it can be, when it dries out, it's quite a fire issue. So we found some along the side of the road, and also at a nursery we checked out, and we work with a nursery. They were really awesome, and they pulled all the stink net out, and we're working with them, follow up, and also talking to the nursery staff, and they're very interested. They don't want to pass this around because it's important for them too. Russian knapweed, biggest area of this is, if you ever want to see it, is over at the corner of Saviors and Hueneme. And one of the things that we did was with this, it's also quite abundant, more than we thought initially. And one of the things about this that's important is that we thought it was something that could potentially be eradicated, but when we started finding it everywhere, we knew that it was no longer possible. So that's kind of the thing that we want to do. We don't want to spend a bunch of money on some of these things that, when it goes up that chart to the right and moves away from being eradicable. So, but what we did with this, we worked with Santa Barbara, UC Santa Barbara, and they have a biocontrol that we're working with that would help diminish the strength of Russian knapweed, but it's not going to eradicate it, but it'll make it so it won't spread as much. That's the idea. So tropical pokeweed, this one, On the right is the American pokeweed. It's mostly a plant that's east of the Rockies. Essentially, it's native to quite common in all of United States, but mostly in the East. In California, it's not. It's more of a weed, but it's not rated. So one of the things, though, is that it is poisonous, and we're starting to see it more often. Tropical pokeweed, on the other hand, which is a little different, and it has a slightly we found it in a field over in Oxnard, and we worked with a landowner, and they're really cooperative, and they just talking to them, and then we work with them, and they removed it. So this one is the one we're after. The American is a problem, and it's something probably people should be looking out because it is poisonous, and something to think about. So this one, onion weed, came after the fire. This one is very explosively expanding. It's a pretty little kind of onion weed looking, like the leaves and such, but it is expanding a lot, and it is considered very invasive. And we're I would say in the last couple of years we've seen it like triple in size, and it is probably not eradicable, but we're working with partners like the Caltrans to remove it. So, and carnation spurge, this one is also in Los Angeles County, is really common, especially toward Malibu, and it's moving into the Santa Monica Mountains and moving less common in Ventura County. Santa Monica Mountains are very common, but again, we were wondering whether we could eradicate it, but having done a lot of mapping and getting a lot of hits on iNaturalist, like people randomly would find this, we would check it out, and then we would see that it was very much more common than we thought. Cape ivy, this one is pretty limited to in our area to the coast. Up north, it's much more common. There is a biocontrol, and you see on the right, there's like that they're trying to use, but it usually stays near the coast in creeks. And but it does take over riparian areas, but it is not such a huge weed in our area. Spanish broom, again, Caltrans has done a really good job on Highway 33. We're trying to remove it, and it is a moderate, it's something that takes up a lot of space and is a huge problem. Tamarisk, we see this various places in our county and in riparian areas, and I think that it's, again, one of these things that ups in our iNaturalist work and the work that was done earlier. Upstream is something that may be more likely to be an area that people can be removing it. And then, of course, arundo. One of the things with arundo that we're seeing a lot is fire in encampments. We're seeing that quite a bit, and it is our number one weed. It is probably it's not in the kind of eradicable section on the left, but it is so important that we really need to work together to deal with it. So stinknet, this is a newer weed. It's kind of toxic to smell, and it has a sort of a nice smell, kind of a eucalyptus kind of smell, but also has also strange chemicals that also will give you a headache, and some people will get rashes when they try to pick it up. But right now you'll see this like a little Christmas tree kind of thing. And finally, I wanted to tell you that we have been working with our ideas to try to get as many entities that are iNaturalist is a super easy app to use, and so we're trying to get groups that are involved out in the field. But so we're working with REI, or we went to REI and gave a presentation. We gave a presentation to California Native Plant Society, the Ventura County Parks, and the other one, the big one, was at Caltrans. They have over 300 people that do trainings, and we had those people because those are the people that are often the first people that are going to see a weed are these workers that are on the ground along the side of the road, and they don't need to be a botanist to work with iNaturalist. You just need to have the app and just be interested in seeing something different, and that could be a useful thing that I try to emphasize a lot. So anyway, one of the things that we're doing in the future that we've done with our partners is at the Ag History Museum, we've had a weed and wildflower show. And we're having another one April 20th at the Ag History Museum with our Ventura County Weed Management Area partners. So anyway, if you're interested in us giving presentation to your group or any information, you can check in with me, John Bell, or Taylor Wallace also is another person that's working on this. Okay.
Thank you.
So let's see.
Because we are a little bit over time, really quickly, we have time for like one question, if anyone wants to say anything. Well, John, thank you for your presentation. I do use iNaturalist, but I never thought to use it for weeds. I mostly used it to try and identify native plants. So I will be taking more pictures of invasive plants now.
Yeah, it really is something that random people and the number of people that are using it is amazing. And but it can be a useful tool if it's free. So if you wanted to have, like, let's say you have a park or open space that is particularly one of the things that we're thinking about is working with some of our partners and including parks. We were talking about this, is like going to some of the parks that maybe are not forgotten somewhat, like I think there's one that are kind of like an iNaturalist desert that don't have a lot of information, and we can kind of like, you can collect information really easy and cheaply through this. So anyway, thank you.
Thank you so much. I know we're pretty much out of time, so I'm gonna just really quickly just tell you guys what my updates are to conclude our meeting. I just wanted to let all the funding partners know that invoices were sent out last week and the remainder will be sent out this week. You're not going to see it come from my email. It's going to come from the executive director of the Ventura County Resource Conservation District, Deborah Gillis. And, if we don't hear with you, I'll probably follow up in a couple weeks. I'm still working with Laura and Public Works on just transferring the website. The current website is on an old system, and I am making room on a Google Drive to kind of hold as many files as necessary. And to do that, I'm moving a lot of the past recordings onto our YouTube channel. So if you go on YouTube and you see like a bunch of past recordings, you didn't miss like a symposium or anything. It's just me transferring some of those files over there to make room on Google Drive for transferring website data. The other thing that might be of interest to you is the state did release a draft of the state water plan. The comment period is over, but it's definitely worth a read. I'm still kind of making my way through it. It's over 300 pages. There's a lot of talk about using watershed networks to build climate resilience, and why they think looking at like watershed scale is the most appropriate venue for planning climate resilience. So I do recommend looking at that, and I'm going to kind of be using that to kind of inform what kind of like structures, like how the group should be structured if we do want to go after some of the funding that DWR has said they're interested in putting out, like a new pilot grant program for watershed networks, and trying to fun studies and stuff for climate resilience. So I'll send those links to you guys in the recording email. And yeah, that's my update for you all. And our next meeting, we won't have a meeting in December, so we're going to skip the December meeting and we will have a meeting in February. Any last questions or anything like that?
Just wishing everyone happy holidays, since we won't see you till February, and some really great presentations. I know today was really jam-packed, but Heidi, thank you for your leadership in putting this all together. EJ, really appreciate you co-chairing, and just thank you to everyone. Really appreciate it. Have a good one.